Okay, we're here today with Susan Bookheimer as part of the Oral Histories series for OHBM. So thank you very much for joining us, Susan.
Thank you so much for having me.
So I'd like to start by asking you about your background and how you became interested in neuroimaging.
It was kind of an accident. I'm a neuropsychologist by training, and I was doing my postdoctoral fellowship at the NIH. So I was in the epilepsy research group, and my job there was to do pre-surgical mapping for patients who were going to get one of their part of their brains removed, usually their hippocampus. And so in that work, what we would do was essentially take a big skull flap off of the brain, sometimes off of both sides of the brain, do direct cortical stimulation of the brain to try to find out where language is. And in a second procedure, we would inject amobarbital into the femoral artery, put one half of the brain to sleep to see if the other hemisphere of the brain could support memory. And of course, this was an extremely invasive procedure with a risk of morbidity and mortality. And the shocking thing about it is, of course, it's a diagnostic procedure. So you could do all of that and get to the end of it and then say, I'm sorry, you can't have surgery because your seizures are coming from where your language areas are. Okay. So I was fortunate enough to be at the NIH when they had this brand new thing called PET, water PET in particular. So they had had PET scanning before, but it was with FDG, fluorodeoxyglucose, which was not ideal for doing functional mapping. But Steve Peterson's paper on language had just come out using PET activation on a very simple word generation task. So we thought, well, why not do this kind of mapping in these patients who were going to have brain surgery? And it would be oh so easy. We would just have them do a language task. We would quickly identify the two language areas that we all learned about in school, Broca's area, Wernicke's area. And we'd be able to avoid all this invasive procedures. So that's what we set out to do. And of course, we instantly found out that everything we thought we knew about language was completely naive and basically wrong. Right. So I spent a few years doing PET work and doing a lot of very, very basic experiments in language just to understand simple concepts like is there a final common pathway in oral speech, and what's the difference in language processing between written language and pictorial language and auditory language. And it was very painstaking, and of course it involved radiation, so we were all injecting our patients with radiation. We used to take turns in those days giving the actual injection so that no one of us would become completely irradiated. So that was fun. And I was just very fortunate to be there when fMRI was first invented, and we had a good scanner at the NIH, and there were some great people there. Peter Gizzard was there, and Bob Turner was there, and Denis Lebien was there. And Jack Beliveau and Bruce Rosen and gang from Harvard came down, and we had a big workshop, and they scanned on our scanners, and we started doing clinical mapping with fMRI. And then from then on, there was just no question that that was going to be the rest of my career.
And what do you see happening with neuroimaging in the US?
Today? Well, there are so many things that are happening in neuroimaging today, and I think things are happening so quickly it's almost hard to keep up with. But I would say that the most exciting things are the big data movements. That is, there are so many opportunities now for getting very, very large data sets, not just of imaging, all different kinds of imaging, diffusion imaging, resting state imaging, as well as, I think, increasingly there'll be some large data sets of activation imaging, but also getting phenotyping along with that, including genotyping. So I think that with the ability to get a rich genetic data set, a rich phenotypic data set, along with these great imaging measures, is really giving us an unprecedented ability to really understand the brain so much better. I think that is the new trend, and of course all of the analytic techniques that go along with that take advantage of those giant data sets.
Yeah. And so what research or other contributions are you most proud of in your career?
Well, I guess I would say there are two things generally, and I'm not one to be very prideful. It's not my nature, so it's hard to answer that question. But I would say that from the beginning of my career, I've been interested in clinical applications, not so much any particular disorder, but in using these really powerful imaging tools to learn more about patient states and how to affect people. So I think that early on we were all interested in basic science and learning basic mechanisms of cognitive science, and I still do that, and I still think it's very important. But I've always tried to steer my career towards having clinical relevance, and I think that I was, I hope to think that I was influential in trying to get other people to do that. But I think in a more concrete way, I think that the most important paper I think I wrote was my I think it was 2000 New England Journal of Medicine paper, where we took individuals, normal individuals who varied on their possession of the ApoE4 allele, which is a common risk allele for Alzheimer's disease, and decided to do activation imaging in normal volunteers who had a different common polymorphism that was a risk. And to our, I think, shock and surprise, in these very healthy individuals who had no signs of cognitive impairment and in fact didn't even know that they had the gene, there were very significant differences in brain activation. And so that got big press and ultimately evolved, and ultimately imaging genetics is now a big thing. So I would say that's the thing that I'm most proud of in my career.
Fantastic. And so you played a part in the creation of OHBM. What was that like, and what did you imagine OHBM would be like?
Yes, I'm not sure that I personally played a part in the creation of it. I was at the first OHBM. I actually gave the last talk at the first OHBM. I developed laryngitis, so I barely squeaked out my talk, and then didn't speak for about 3 days after that. But it was a very exciting time, and it was a time of great and active discussion about whether or not we should create an organization or whether this should just be an annual meeting. And it was a very controversial topic. Others will be much better at speaking to you about that. It was amusing, actually. But the truth was that we really needed OHBM. Before that, before fMRI, we were PET investigators, and the only meeting that really made sense for us to go to was cerebral blood flow and metabolism, because we were measuring, in fact, cerebral blood flow. So we joined this meeting with all these other people who were studying basic mechanisms of blood flow and metabolism, mostly in animals. We used to call them the metabolists. And then there was this completely separate group of individuals at this meeting who were doing activation imaging, and the metabolists had no idea what we were doing, and we had no idea what they were doing. So it was really not much of a happy marriage. And so it was necessary to have another organization that really appreciated brain mapping per se, human brain mapping, the idea of relating brain function to brain structure. So many of us were involved, and it was Bernard, of course, who put together this first meeting, and then a bunch of individuals, including Peter Fox and John Mazziota and Alan Evans and many others, who Richard Prokowiak, who pushed forward the idea that we should have at least a meeting, and maybe it should be an organization, and now I think we're going to become a society. Mm-hmm. It was a very exciting time.
Fantastic. And what have you found the most rewarding about your involvement with OHBM? I think that this, first of all, scientifically is an amazing meeting. It is always so exciting. There are always new techniques, new acquisition techniques, new analysis, and of course new applications. So I don't think I've ever been to a meeting here where there wasn't brand new science that was really exciting. I think that's one of the things that's most extraordinary about this meeting. Of course, on a more practical level, I've been with these people now for 25, almost 30 years. We've all known each other, and so it's a great opportunity to see friends and network and just watch this field evolve. It's been so much fun. And so what memories stand out in particular when you think about your experiences with OHBM?
Seeing Alan Evans at the annual town hall meeting with a combat helmet on, that was one of my favorites. There's so many fun things. And early on we used to have parties between the Los Angeles group and the London group that was Hammersmith back then. We used to call it the L&L party. And that grew and grew and grew, and of course ultimately evolved into the party night that we have now, the club night. But it used to be a private party, and we've had so many fun parties over the years.
And what changes have you seen with OHBM over the years?
I think that there are many things that have changed. Obviously, the evolution of the science is the most exciting thing, but we've become so much broader in so many ways. The involvement of the younger people has been especially exciting. I think the special interest groups, the COBIDAS group, all the big data groups, there are so many new initiatives by the young members. That is new, and every year it's new. It gets more and more exciting to see the replication award. I think is a great idea, really unique in science, honestly. I mean, I could go on and on. I think we really do continue to evolve, and I think that's one of the great things about this organization.
And lastly, more broadly, what do you see as the future for neuroimaging?
It's so hard to predict the future because I think, when I think back in the late 80s, that we never imagined that we would be doing what we're doing today. Doing neuroimaging back then was so painstaking, so time-consuming. We didn't have these SPM and FSL and ways of analyzing data very quickly. We did everything manually, and it seemed like such a chore, and that it would always be a small group of people doing this because it took so long and so much energy. And now we just have the opportunity to grow incredibly because we have these incredible data analytic tools. I think that's one thing that's changing. One of the things that I'd really like to see happening, and I do believe is happening, is that I believe that brain imaging generally is going to be incorporated more and more in clinical science. I think that looking at brain structure, brain function, even resting state function, as the dependent measures or even outcome measures of clinical trials is going to increasingly become the norm. I think that the current state of the art, if you can call it that, in clinical trials, to try a new intervention and wait to see if somebody develops a bad disease and dies, for example, is a really bad way to conduct a clinical trial. And imaging has the opportunity to change that dramatically and look at markers that are so early and also so much more closely related to the biology of these disorders, that I think that we're going to become very integral to clinical science in the future.
Professor Bookheimer, thank you very much.
You're very welcome.